when I told my American sister-in-law that I was moving to the hospital in Salzburg, she directly said, "Oh, Salzburg, this is the sound of music and we love it." I did not know that before, but this is why I choose this picture as an introduction. I'm also not a specialist on Barth syndrome, and I hear that you are all scientists and I'm a pediatrician, so forgive me that my presentation is perhaps a bit more clinical and that I perhaps do not know the really tiny biochemical details, but we will find a way. I'm interested in a biomarker you can find in the urine, and this is 3-methylglutaconic acid. And when you look at these pictogram, it's a pictogram of all metabolic pathways in the human, and you see this can be quite confusing. And these marker, these 3-methylglutaconic acid, can help you a lot because it can help you to recognize patterns and to make clinical diagnosis. And this is the growing family of these disorders. They are all characterized by 3-methylglutaconic aciduria, and I will talk about this marker a bit more in the next slides, in the next minutes. And as you see here, and as you all know, Barth syndrome is also characterized by this biomarker, and there are several other disorders. And my research was focused on looking what does this biomarker do, where does it come from, are there any other disorders, can we find a better classification, because it was quite a mess when I started this work. And today I will focus on the disorders which we found out play a role in the phospholipid metabolism and not focus on these other ones. But as a short introduction about this 3-methylglutaconic acid, everyone always asks, where does it come from, where does it stem from, and we really do not know it. We only know it for the what was formerly called the 3-methylglutaconic aciduria type 1. This is really a classical inborn error of metabolism in the leucine catabolism, and due to a defect in this enzyme, the 3-methylglutaconyl coenzyme A hydratase, you get elevated excretion of 3-methylglutaconic acid, 3-methylglutaric acid, and what discriminates this disorder from all the other 3-MGA syndromes is that you have an elevated excretion of 3-hydroxyisovaleric acid. For a long time, people thought that this might be a non-disease because there were only several patients described with quite different clinical signs and symptoms with not a specific pattern. It were all children with speech delay or global developmental delay, but then we could show that it is an adult onset disease, and with these adult symptoms are very specific. And you can see here on the MRI an impressive leukoencephalopathy, and these patients develop a progressive spasticity and dementia, but only about the age of 40 to 50 years. So this is certainly not a non-disease, but a very special disease with an adult presentation. This leucine pathway plays a role in these type which I already showed you, and I investigated in all the other types if it could stem from these leucine catabolism, and this is not the case. These studies were earlier done for Barth syndrome as well, that you measure the 3-MGA before and after leucine loading, and you do not see any difference in the 3-MGA excretion before and after for all these other types of 3-MGA syndromes, only for the type I just showed you. So I think we can state that it does not come from the leucine catabolism, but we really do not know where it stems from, but it seems that all these disorders who show this as a marker have some pathomechanism in the phospholipids or in the mitochondrial membranes. This is a classification I made for this. I think this is not scope of my talk today, But be careful, you can also find this 3-methylglutaconic acid generally in very sick children or in presentation of other metabolic disorders. And this is the classification based on the pathomechanism. So I will talk today about these defective phospholipid remodeling disorders and not on the other types. In 2006, I described 4 patients with a disorder we called Mechdel syndrome, based on the acronym of the most distinctive features. And this was the 3-methylglutaconic acid. They all had the very rare clinical symptom of dystonia combined with deafness, and they had an encephalopathy and a Lee-like syndrome on the MRI. I will come back later to that. We also at that time had 1 patient who had a neonatal liver failure. We were unsure if this is part of the disease or if it was just an infection in the neonatal period. But later it turned out that this liver failure is also a typical sign of Mechdel syndrome. These are some photos of the patients. By now, I know 75 patients who are in my database, and I would like to point out some of the typical clinical features to you. This is a boy from Croatia who had this neonatal liver failure, and you can see here the hypotonia in the rump, the spasticity in the legs, and the dystonic movements of the hands. What you cannot see is that he is deaf and does not react to acoustic stimuli. I think this hypotonia, what you can see in him, you can also see very well in this patient. She has to lean on the couch because otherwise she cannot sit by herself. You see the spasticity of the limbs and again these dystonic movements. And it's a progressive disease. This is her 14 years later. And I think also very impressive for the progressiveness of this disease is this boy from Afghanistan. Here, he's quite normal at the age of 3 years, and you can see here the progressive spasticity, which we could not treat with any medication that was available in his country in 2009. This girl is a girl from South Africa, and she was a little later with the presentation of symptoms. You can see her here in the early infancy. She learned to walk, what most of the patients do not do, and you perhaps see a bit of the spasticity in the legs, but still no big problems. And this is her, I think, a month ago. She's now aged nearly 8 years, and you can see the severe spasticity, the severe dystonic movements, also the dystonic movements periorally, and she's currently, because she has lots of problems with infections, in a very bad clinical situation. Regina, may I see the video? Woo. For me, this video of a little boy from Warsaw in Poland shows all the features I described to you. Again, very good in 1 patient, and this is the typical presentation you see in all patients. I want to draw your attention to his mouth because you see him here with this continuously protrusion of the tongue. I think this is also very typical for these patients and the reason why they have so many problems with hypersalivation. These are some pictures of the faces of the patients, and you might see some kind of overlapping dysmorphic features, but I think these are not primary dysmorphic features, but they are secondary due to the course of disease, the same as you see like a mask-like appearance in Parkinson patients. So I think it's not a primary dysmorphic feature, but a secondary feature of the neurological symptoms. Here are some figures from a liver biopsy of the patient. The patient, or you can find OXPHOS dysfunction in a different Cox staining in these cells in the light microscopy, and in the electron microscopy of the liver, we saw different things, and I think the best described, they were not very typical, but with damaged mitochondria. What is also very special about this disorder, all these patients show exactly the same course of disease, exactly the same ages where the symptoms appear, and you have a very specific MRI pattern. You first see a swelling of the caudate nucleus and the pallidum in the first year of life, and then at the age of 1.5 to 3 years, you see what we call the putamen eye. This is in the basal ganglia, in the mid-dorsal putamen, a spared region. And this seems so pathognomic for Meckel syndrome that we even diagnosed patients only based upon the MRI. After I showed this on a meeting, people came to me and said, oh, I saw these MRI, and we were really questioning what this is, and we checked for SERAC1 mutation, and it was the case. The clinical course, I have to update this picture. This is on the first 54 patients, is that you see this 3-methylglutaconic acid. You see it during the whole life. We only found 2 patients who did not show this marker. All patients have deafness, and the patients do not learn to speak. Some of them are babbling, some of them learn some words, but they lose it again. Many patients have neonatal liver problems. There are even some patients described who did not survive this neonatal liver failure. And it's very interesting that if they do not have these problems in infancy, they do not, you do not see these problems later in life. So it's really only a neonatal feature, and I cannot understand it based on the pathomechanism. What you often see is that they have transaminases going up and going down, but no clinical correlate of this. The patients show a severe developmental delay. Some of them learned to walk, but most of them never learned to walk and all lose it again. They show this progressive spasticity, the dystonia, and most of them also show severest drooling. And they have these typical MRI findings. So I'm happy that with the acronym we choose, that also with investigating more patients, that we choose the right acronym. Concerning the survival, I'm quite surprised that they survive long. The oldest patients are now into their mid-20s. And if they die, you have these 2 age periods, it's due to respiratory infections. I think this is a combination of the hypotonia of the rump, the scoliosis, and this hypersalivation leading to aspiration pneumonia. But why I am here, and what I'm very happy to discuss with you, because I need your help for this disease, is the underlying pathomechanism. When we found these patients, we thought there is some overlap with Barth syndrome. We have the same 3-methylglutaconic aciduria, we have OXPHOS dysfunction, and many of the patients also have a low plasma cholesterol. So what we did, we contacted Frederick Wass and Ronald Wanders in Amsterdam and asked them to measure the phospholipids. And I'm now going a bit too fast. I have to show you the gene first. We did exome sequencing on 2 patients, and we were very lucky that we only found mutations in both patients in 1 gene. This was SERAC1, a gene of unknown function, and the only thing we knew was that there was a lipase domain. We could find mutations for all these patients. There's no genotype-phenotype correlation, and there's also no hotspot region. In this gene, the mutations can be in the complete range of the gene. But now for the phospholipid metabolism, why I'm here. Frederick Wass and Ronald Wanders measured the phospholipids, and what we found was very interesting. We found elevated levels of phosphatidylglycerol with the 16:0:18:1 side chains. We found diminished levels of the PG 18:0:18:1 levels, and we found diminished levels of BMP. So we thought that this is a new pathway, because the pathway towards the BMP was not known. And what we think what the function of SERAG1 is that it cuts off one side chain and puts again another in the place. And to be honest, I'm still surprised until now how such a tiny, tiny little mistake can lead to all these clinical signs and symptoms. The cardiolipin levels in these patients, or this PG, is one of the substrates for the cardiolipin synthase, and then you get cardiolipin and the remodeling. I don't have to tell you about that. So we were interested in the link towards cardiolipin. So we measured the total cardiolipin amount in the patient cells, which was normal compared between patients and control cells. And in the next step, we were interested in looking at the cardiolipin subspecies. For comparison here, in Barth syndrome, the total amount of cardiolipin is low. And in Meckel syndrome, we have a normal amount of cardiolipin. But what we saw is that the cardiolipin subspecies, which derive from the PG34-1, are elevated in patients. And we think that this is due to, this is not working. So you have an excess of these PG34-1 and low PG36-1. So the cardiolipin subspecies coming from here are elevated in the patients. What the consequence of this is, we really don't know. But we think, as many of our patients show OXPHOS dysfunction, some in muscle, also some in fibroblasts, some only in the ATP production, some in different complex activities, that this could be the link to the OXPHOS dysfunction, but we do not know details. What is the subcellular localization of SERAG1? Of course, we first thought that it is a mitochondrial enzyme, and we found it in the crude mitochondrial fraction with this marker NDUFS3. But then we had a closer look, and we checked also for the ER, and we found in both, we found the same expression as for calnexin. So we think that SERAG1 is in this MAM fraction, the mitochondria-associated membrane fraction, and I think this would fit very well because this is the fraction where the phospholipid exchange takes place. Another very interesting feature of Meckel syndrome is that it is not only the second known phospholipid remodeling disease, but also the second known cholesterol trafficking disorder. Because of the low BMP, we studied the Philippine staining in these cells. And this is the positive Philippine staining in a Meckel patient. And this is the positive Philippine staining of a patient with a disease you might know. This is Niemann-Pick C disease. And this is the healthy control. So it is known that if you have a low BMP intracellularly, that the cholesterol is trapped in the cell, and then you get this positive Philippine staining, what we see in our patients and in Niemann-Pick C patients. If this is the reason for the low serum cholesterol, I don't know, I can only speculate. But I think it's very interesting that both patient groups show an overlap of the clinical features again, because Niemann-Pick C patients also have neonatal liver problems. So perhaps this is the link to the neonatal liver problems, I don't know. So taken together, Meckel syndrome is a highly characteristic clinical syndrome with a highly characteristic radiological phenotype, and it is the second, or was at that time, we found it in 2012, the second described phospholipid remodeling defect, and the second known cholesterol trafficking disorder. There's at the moment much interested in these disorders. They are claimed or named by Professor Sotobre from Paris. He was the first to see this. A new class of diseases, the diseases or defects in the biosynthesis and remodeling of complex lipids. And I made this figure for an article in the Journal of Inherited Metabolic Diseases. And what you can see here, in the cardiolipin synthesis pathway, there are in fact 3 disorders. This one you all know very well. This one you just heard about. And there's another enzyme. This is the acylglycerol kinase, which underlies the so-called Sanger syndrome. And I want to talk about that as well for a short moment. This is a patient from me at that age, or at that time, she was 10 years old. And what you don't see in her, she's a very active and happy girl, is that this is her heart MRI from the same day. So she has a really, really impressive hypertrophic cardiomyopathy with so-called kissing ventricles. So the radiologist phoned me after this and really asked, how is this patient doing? And she was just in the outpatient department and was springing around. Because here there's nearly any space left for the blood to circulate. She has cataracts, have been operated when she was very young. She's doing quite well. She goes to a regular school. She had a brother who sadly died when he was 15 years old due to rhythm abnormalities. He had a sudden cardiac death. And if you ask me, is she the typical patient? Yes and no. We know some patients who are already adults and have a quite mild form of disease, but we also know many neonates who do not survive the first 3 months. So this is a disorder with a broad clinical spectrum. What is interesting is that also for isolated cataracts, AGK has been defined as a candidate gene or as a disease gene. So it seems that you can have a very mild type of the disease with only cataracts. This is my colleague from the lab in Salzburg. This is Hans Meyer, who was for a long time interested in Sanger syndrome and also solved the genetic background. And unfortunately, we were unable until now to exactly measure the phospholipid profiles in these patients. But based on what is known about phospholipids, AGK should play a role at 2 stages in the reaction from monoacylglycerol to lysophosphatidic acid, and in the in this reaction between phosphatidic acid and deacylglycerol. So it is thought that Sanger syndrome has an effect on the phospholipids and on the membrane composition of mitochondria, but also via this way you are going into the signaling, into the IP3 signaling, so that it could that could also play a role in the pathomechanism of this disease. There was a long time speculation that Sanger syndrome is caused due to mutations in ANT, the adenine nucleotide translocator. This was not the case, but they found out that AGK is involved in the post-translational modification of ANT. Just very briefly about one other disease. This is DCMA syndrome due to mutations in DNA GC19. This has been first described in a large Hutterid family with dilated cardiomyopathy, cerebellar ataxia, testicular dysgenesis, and growth failure. Also here, many patients did not survive the first 5 years, but there are also patients with a more attenuated phenotype that are older. This was published in 2013, and since then only one Finnish case has been published, and 2 months ago we found one patient from Turkey. It is not exactly known what is the function of DNA C19, or there's a bit discussion about it. It shows a high homology with TIM23, a protein that is involved in the mitochondrial import and which is mutated in Moore-Tranebjerg syndrome. This is a syndrome characterized by dystonia and deafness, so we are still in the same loop of the clinical signs and symptoms. But there's also a study from Richter-Dennerlein from the Thomas Langer group in Cologne, who found that DNJC19 forms a complex with the so-called prohibitins, and they state that cardiolipin remodeling is dependent on these complex, so they state that this is also a phospholipid remodeling problem. I don't know, I discussed this with Frederick Voss and he was not very convinced about this. I don't know, I think there are more studies to follow on this. So this was what I wanted to show you. I think that 3-methylglutaconic aciduria is a very good marker to find all these different disorders. And for me, it's a fascinating pathomechanism, but you have the favor of a big society and a very active society that is interested in research on this. But until now, I'm quite alone with my research on Meckel syndrome. I already found 75 patients, so I think it's not a very, very orphan disease. And I'm very happy to be here, and I would be very happy to find collaborators in the field to more work on these disorders, especially on Meckel syndrome. Thank you very much.
Great, thanks, Saskia. Since we're running a little behind, we have just maybe one question.
So this was very interesting. And I have a question regarding the SERC enzyme and its actual function. I know that with you and Ron showed that this, the PG molecular species pattern changes, and that's, of course, very interesting. But you know, if it has something to do with the BMP pathway, I think that sort of would make a lot of sense, right? The BMP pathway actually is not very well understood. And yes, PG is a substrate, but a lot of things happen along the way, one of which is that the stereochemistry changes. So the head group flips over, which is something nobody has been able to figure out. So I'm wondering whether this enzyme or the reacylation, perhaps deacylation of PG has anything to do with that, with the mechanism of this stereochemical flip.
Yeah, yeah.
I mean, this would be very interesting to study. You have the perfect model for it.
Okay, great. Thank you so much.
Thank you.